Okay.
Wait, what was it? Well, how was it?
How's everybody doing? I'm also good. I'm just making sure that everything's working with the recording and then we can get started. Can people online hear me and see my slides? Yes. Okay, awesome. Okay, so today I'm Tyler Morgan. I'm a staff scientist in the fMRI core facility here at NIH. And we're going to be talking today about layer fMRI and columnar fMRI. And okay, everybody seems to be on. Okay, so we're just going to start with what I would kind of think in this topic is a pretty easy definition. Functional fMRI has provided a lot of really interesting information that tells us about kind of the cognitive preferences of different places on the cortex. We can kind of think of this broadly as just brain mapping in general, where we've spent a really long time being cartographers of the brain, and we basically are like, oh, what is this task? What does it do, etc. What kind of processes are involved, and then seeing which areas of the brain are associated with that. And that's given us really cool types of information about the brain, things like retinotopy, which is like a topographic organization that's associated with how we see vision or how we associate vision. But you can also have kind of crazy things like task-induced suppression. So we had a little bit of a talk about this earlier in the week that we heard about suppression in the default mode network. That's what we're showing here. But it can get really complex really, really quickly. So we went over this in the deep scanning or deep sampling lecture a few weeks ago. But this is a study where basically people listen to podcasts and audiobooks for a very, very long time. And then they took models of all of the concepts that were being talked about during the length of people being in the scanner. And then they mapped which parts of the brain were associated with those types of concepts. And then they used a whole bunch of algorithms to try to figure out where in the brain are those things happening. But it's, I think, really important to remember as you get deep into the neuroscience of how connections are in the brain, you need to remember that it's not just a single flat sheet, which is how we're showing it here. Actually, it's made up of a whole bunch of different layers, and those layers are in turn also made up of different types of cells, different types of connections, etc. And so we can totally imagine that these might be really important things for how the brain functions. If everywhere on cortex seems to be organized very similarly, or relatively similarly, and the cells in each of those layers are kind of unique, then it must be doing something important. What is also really interesting is that when we start looking at really high resolution fMRI, we start to observe different types of activation or different layer profiles when we look at different types of tasks. So an example here is like people tapping versus their fingers being brushed or having piezo stimulation. Those don't look the same. They actually, some of them have different layers that are associated with them or different patterns where different layers are kind of stronger or weaker. And we can ask very easily, like, why does this happen? What is it that really causes this? And what's the neuroscientific backbone of why this happens? And I would say that that's really our definition of layer fMRI, is the study of not just where in the brain that we care about, but also why this particular organization happens in cortex. Do you mind closing that door? Just because of that sound. But yeah, one of the reasons, and I think a really important reason, I already touched on it a little bit, is that there are a whole bunch of different types of cells. So even in this particular case where we're only really showing, well, we're showing quite a few different types of cells here, but they are mostly associated with like It's just never gonna go away. I think so, yeah. These are all mostly pyramidal action cells, so basically you can see that there are a whole bunch of different types and a whole bunch of different depths, and also the structure of those cells is very different. You can think of some of them as being very localized, like we have here, but you also have some where their connections are actually pretty much in all of the different layers. It turns out that most of them are kind of concentrated in a few different layers, but you can see that they span this entire structure. And the other really interesting thing, I think, is that we can also look at the type of receptors that exist in each of those. So not just the cells, but also the specific chemicals and the specific types of actions that those neurons take are associated with different chemicals that they're sensitive to. So you can see some examples here like GABA, AMPA, NMDA, et cetera. And those all have different concentrations that concentrate differently in different layers. So again, just an example of why we might consider certain types of activity happening in certain layers as being important. And even beyond that, I mean, everything that I've shown you is basically just these cells are different, but that doesn't tell us how they are actually organized. And we are currently, I would say, going over the last 30 years or so, we have gone through a lot of understanding and kind of revolution in our understanding of how these different cells are connected and what that might mean for computation in the brain. So a particular brain area receives information. It also sends information to other places, but how that happens and what information is processed while it's in that area, that's a really interesting question. Here we can see it just looks very, very complex, I would say. But we can also see that there are some interesting patterns that happen here as well. So for instance, I don't know if everyone can see all the details of this figure, but we can have 3 different cortical areas. And what we're actually looking at here is how are different cells wired together to different areas. So how are they passing information to different stages of processing when it comes to the brain? Okay, so all of this kind of sets us up for this really cool part of the field, which is that we have neuroscience that has been concentrating for a very long time on the molecular and the cellular levels. And we are gaining methods over and over again that actually increase the field of view. They make it so that there can be different types of invasive methods. And we're building up to being able to see not just the cellular view, the microscopic view, but also the mesoscopic view. So this is the in-between scale in Greek, I guess. At the other end, a lot of us come from the fMRI world or the MRI world in general, and that really starts at the macro scale. So you're looking at networks, you're looking at individuals as their entire brain is doing something, or within areas they're doing something, and we're working our way backwards. So we're working our way to more and more detail. And those things are kind of meeting in the middle in a really cool way where actually we're having different types of science be informed by one another that have not really seen each other in the past. So that is what I would call mesoscopic, mesoscale imaging. And the interesting thing about it is that it encompasses both layer fMRI, but it also encompasses columnar fMRI that we talked about in the title. We saw this last time, but obviously in order to get high resolution, either layers or columns, we need high resolution machines. And this most of the time takes place in ultra high field MRI machines. At NIH, the 2 that I work with the most are 70A and 70B. For those that have been down to the center, you might know where those are. We also have one other 70 in MRF 70. But these can go much higher, and it becomes more challenging to do it. But the whole goal is, like we said, to push further and further into that mesoscopic or even try to get on the microscopic level. So you have scanners like in Minneapolis, they have a 10.5, it's huge. In 11.7 in Paris, it's even huger. These magnets keep on growing and growing. And we also saw this last week. Essentially, the really nice thing about going to higher fields is that you gain more signal. So the amount of available signal in different parts of the brain grows as you're as your magnets get larger. And the really nice thing is that it's super linear. There have been a lot of debates, as we heard last week, about what are the types of things that actually make this super linear. But for the most part, most of our empirical data shows that you get more bang for your buck as you go to higher fields. So 70 is a nice place to be because essentially we are in kind of a sweet spot where we know how to make them quite well. The technology exists, but we're also getting that increase in SMR. So going to these really crazy machines that there are only a few in the world, that's a big engineering feat in comparison to, you can now just buy a 70. And this is really important. The amount of signal that we get is really important because most of the time when we're doing things like fMRI, and particularly mesoscale fMRI, really what we're doing is we're pushing our resolution dramatically. So rather than summing over all the protons in like a 3x3x3 voxel, we're then splitting it apart into all these little ones that also need to have signal to noise in order for us to be able to do really anything. Okay. So what do we actually expect to see? What's the goal of layer fMRI per se? I would say one of the main things that we really care about is the separation of different signals as they come in from the outside world. So this is feed-forward activity or sensory activity, and the type of activity that is associated with things that are already in your brain. That's a really simplistic way to put it, but basically our internal models of the world are knowing what is outside of that door just from memory. You don't have to go look at it to confirm it. You are quite sure that you know that there is a staircase out there, etc. So these are associated with a type of signaling that is called feedback. And basically what those two streams are doing is constantly interacting. So you have expectations and you have models of what you think the world is like. You come in contact with the world, and the world either throws you for a loop and you have to remodel, or alternatively, it doesn't. Like it just meets your expectations, etc. And there are also a lot of really detrimental situations for the brain when these two things become out of balance. So we can think of examples like schizophrenia, etc., where you might be more influenced by one of those streams than the other. So theories of feed-forward and feedback information can be tested by both separating high-level perception from low-level features, and also in some cases actually removing stimuli in order to test expectations when feed-forward information is not really met the way that we would like it to be. And we can kind of think about this as designing experiments where now when we have really high resolution fMRI, we can look at all of these different layers. I guess I didn't mention that oftentimes we kind of, from a very high level, conceptualize feedback information as traveling through both the superficial and the deep layers of cortex. Oftentimes, as you've probably seen in neuroscience classes that you've taken, then layer 4 or the middle layers is often associated with the information coming into an area, although this isn't always strictly true. But in general, we have the resolution that we can start to see where we would expect these different types of signals to be. And then we can also design experiments that either remove that information or change it in a way that makes it so that we can study it. A kind of cool example of this is a study from Peter Koch. And here, essentially what they did was they looked at a pretty common illusion. It's called the Planisa triangle. I'm sure others have come in contact with it, but it's essentially, it's basically when you have 3 circles and then an overlaid invisible triangle over the top. So there is no edge to the triangle, but almost everyone that sees it actually sees a triangle. And that's what you're seeing in this green section on the top. They have 4 circles in this particular case, but they have them rotated so that on the left-hand side, you can see an overlaid triangle. And then on the bottom case, they rotated the circles again, and it breaks this illusion. And what they wanted to look at was if you looked in visual cortex, could you actually look at information in different cortical layers and understand, is the brain trying to fill in the information that we normally perceive, which is this triangle, in different places? And so they looked at V1, and then these are what are called projections. So basically taking the information from the brain, the activity that's in each of the voxels, and mapping it onto where in the visual field they are actually happening. And then you can take a pattern of brain activity and you can put it on a stimulus in order to understand where it actually was associated with. That makes sense to everybody? Cool. And so then what they're showing here is basically just that in the deep layers, they have a larger amount of activity that is associated with that edge. They're specifically interested in the place where there is no stimulus, it's just blank, but we perceive an illusory stimulus. And they wanted to know, is there actually activity there? And it turns out that they found mostly in deep layers that there is, but in other layers there really wasn't. It was much, much weaker, and it never met statistical thresholding. And so they concluded that part of this illusory information is actually being processed or contained in the deep layers activity. Lars Muckley also did a kind of similar study, I would say. And this is also trying to remove feed-forward information or create situations where there is no information in your sensory domain. But we still see activity that's associated with the things that you're experiencing. And just a note, this is the lab that I did my PhD in, so I'm very acquainted with a lot of these, but basically what we would often do is we would take natural images, so not just illusions, but like very complex images. And we would show them where people could see the full image, or alternatively, they would see it where it was blocked in a quarter of the image. And then what Lars was really interested in one of these early studies was to look at different cortical layers and try to understand whether you actually see activity in the portion of the brain that is associated with this lower right quadrant that's covered up, and see whether you could actually still decode images from one another. And it turns out, yes, you can. But in the cortical layers, you can actually see that in this solid line here, you can't see it in the deep layers. You can only see it in the superficial layers, which is kind of cool. As opposed to when you can see the entire image, you can decode it everywhere. So they interpreted that as upper layers are containing a lot of the information about like complex images and how they might be filled in. And I'll just point out some of the work that I was associated with, because I think it's cool. We did something very similar. We showed images where we could see the whole image, or we could only see a portion of those images, and then we decoded those from one another. But the really nice thing that we could also do is we can actually do this experiment in both humans and in monkeys. And this allows us to not only see from the fMRI point of view, where we can kind of have a good idea of what's happening everywhere in visual cortex, but we can also record from individual cells in the monkeys as they're doing a very similar experiment. And basically what we found we found something very similar in the humans, but in the monkeys, we found basically that there are responses when the images are included in neurons that are in that blocked out portion of the image. But more importantly, we can also decode between, it's 24 different images in this case, and the temporal dynamics of when that information is actually represented in those neurons is different. It's slower, which for those that are not acquainted with it, might be more associated with feedback, because we often think about information coming in through the eyes or through any kind of sensory modality, essentially doing a pass where we get all the information that we can, and then we start making computations on top of it. So this is the feedback information that happens afterward. And I think it'll come into play a little bit more, but I'll just point it out. It'll come into play a little bit more later in the talk, but essentially, you can also look at whether this is associated with that feedback signal that we were talking about, or alternatively, if it's more related to something called lateral connections. Just really quickly, the difference between those two is that they both pass information from different neurons in the brain, either in between areas or within the same areas. But importantly, feedback is most associated with information that's coming from other areas. So if you're in B1 and you're processing some kind of information, you might get information from B2 that's a little bit higher level that tells you exactly what's happening. But those are quite long range connections and they're sparser. Lateral connections, on the other hand, are like how you talk to your neighbors. Like you as a neuron probably are associated with, or you're probably seeing very similar things to your neighbor neuron, etc. And you still probably talk to each other about that, but it doesn't necessarily need that long range connection. And we can actually examine both in monkeys and in humans whether those are the types of information that you're getting. Are you getting that information from long range connections or from short range connections by looking at how information is there more information closer to the border of that blocked out portion? Or actually, is it not really there? Is it just coming everywhere at the same time? And it turns out that it seems to be coming all at the same time. This is kind of an interesting aspect of that, and I'll just cover it really briefly. You can use representational similarity analysis, which we've seen a little bit in the summer course already, in order to compare the data that you get from the two species and see whether that information is being kind of represented the same way. Turns out it is, but on a specific timeline. All right, another example of kind of layer information and how you can look at it is to not look at decoding, etc., the pattern of information, but you can actually just look at the temporal dynamics. This was work that was done by Jinyu in Alan Koretsky's lab at NIH. And basically what they did was they designed a new type of sequence that used saturation pulses in order to get rid of the signal from almost the entire brain, other than a little sliver. This is a mouse brain, in case you're trying to recognize it. And a little sliver, which then they could just look down really, really quickly with the MRI system. And this basically was a departure from the way that we often collect MRI data, which is to cover, those of you that have done it, you kind of cover a large field of view and you try to read out all of the information in that. It's quite a slow process. Here, they wanted to get really high resolution through cortical layers, but they also wanted to get really high temporal resolution. And so here they were able to accomplish 50 micron resolution and 50 millisecond temporal resolution. And what they found was that then they could look at somatosensory cortex in the rodent brain, and they could actually just record regular fMRI, but they could see temporal dynamics that they could never see in other forms of fMRI. So here you have time going this direction, and you have cortical layers here. They're marked as layer 1, layer 4, layer 6, etc. And in the case where they just did a feed-forward stimulation, so in other words, they did a tapping on the foot of the rodent, they could see that the activity started in layer 4. And that's exactly what we would expect if that's just feed-forward information coming in first and then getting integrated across. However, if they went to the other side, so they didn't tap the contralateral, but instead the ipsilateral, but then they started to see something different, which was that the activity is no longer starting in layer 4. It's actually layer 4 is pretty late. But layer 2, 3, and layer 5 are getting that information first and then integrating it with the rest. And the reason that this might happen is when you look at the ipsilateral side, essentially you have information going to the contralateral side, being processed by somatosensory, and then being sent to the other hemisphere. We also did this in humans, which I think it was a huge challenge, and I'll only go really quickly through it, but we basically worked on trying to make it so that, oops, kind of weird. My computer is very slow today for some reason.
But we were doing something very similar. You can see a human brain now, and you can see a line that is just associated with canceling everything else out, which is hard because human brains are much bigger than rodent brains. And then looking at a very, very specific place on cortex, which you can see here on the 3D model of the cortex, in order to understand a little bit better the properties that are being processed there. And so one example of this was done in somatosensory cortex. And we just did something that was relatively similar, I would say, to what they did in rodents. So here in the contralateral side, you can see that there's a lot of activity at the very top of cortex. This is a well-known phenomenon called superficial vein bias, where basically the bold signal is always the biggest at the surface because that's where your biggest veins are. But really, oftentimes we care about the stuff that's also happening down below. But the contralateral side is, again, the side that you expect the most activity with. It's where feed forward is coming in first, etc. So then if you switch to the other side and you looked at the ipsilateral activity, you can see that the pattern is very, very different. It's actually negative, which is already an interesting thing in fMRI, but also it's not negative in the same places everywhere. So therefore it's actually, or this is motor. So therefore is not actually reacting nearly as much in that other side of the brain, but the rest of them are going negative. So that's already interesting in itself. At the same time, you can also look at higher level information that's being shown. So here on the left-hand side is basically monkey data that shows that throughout V1, you have different tuning properties for the neurons in each of those layers. So in other words, how much of the world they see, which is called the receptive field size, and what they care about, the orientation of lines in the world in vision that they can see, change over these different layers. And in both cases, so RF size, which is the size of the amount of the world that it can see, it gets smaller as you get towards the middle of cortex. And also, it seems that they don't care about orientation nearly as much in the middle of the cortex either. But they get larger as you go out, and you can see that they care about orientation as well. And we repeated a very similar experiment in humans using our method, and it turns out that you see something kind of similar. It's not exactly the same, but I would say this is one of the really exciting parts about LAIR fMRI is, like I said, this interaction between different fields that haven't traditionally been able to talk to one another very well. Okay. So what then is, what are cortical columns then? Well, like I said at the beginning, this is kind of the other functional unit that we really care about in mesoscopic fMRI. And basically the cortex itself is kind of divvied up into these little tiny patches. And those little tiny patches, again, talk to one another, and they talk to one another through feed forward information, lateral connections, as well as through feedback information. And they're small. In monkeys, they're about 200 microns across for what's called a microcolumn. The macrocolumn is maybe approximately 3 millimeters or so. And in humans, they're a little bit larger than this, but not dramatically. So columns are small functional units that span all of the different cortical layers. And like I said, they're connected to each other through a whole bunch of different types of connections. Now, when it comes to fMRI, one of the interesting things is how do you find these columns? Because to us, in both MRI and also if you're literally just looking at a human brain, it just looks like a giant sheet. You can't really know where each of the little units, the functional units of the system are. They're not like circled or anything like that. It would be great if they were. But so we have to think about how we actually hunt them down. And there have been a couple of different methods for how to do this. One of the early ones that I'm aware of is actually just looking at a functional property that you care about. So here they're looking at tonotopy. So what kind of frequencies do we hear? And they're looking for places in cortex where all of the different layers, all the voxels throughout the layers of cortex care about the same frequency. And that's, as you can imagine, not uncommon, but it's not constant. It's not everywhere. And I'm showing a few examples here just so that you can get an idea of what that means. So here you can see that they change throughout the different layers. That means that that's one column. The same here. But if we go down to the bottom row, we can see two examples where it's red all the way through in this case, or it's blue all the way through in this case. And that's demonstrating that all of the layers of cortex care about the same frequency in this particular little patch. And they're normally quite small patches. And so they would define that as a cortical layer, or sorry, as a cortical column. Alternatively, you can just think about this as like, okay, we're probably never going to get it exactly lined up necessarily, but we can just divvy it up geometrically. So we can take the whole cortex and we can split it up into these little patches that you can see a 3D rendering on the right-hand side there. And basically you just get a little, I mean, this is only a small patch of brain, but you can do this over the entire brain. And then you just get these little sections. They're basically just little Barnoi sections, really, that you can then analyze and try to understand how things are changing in the brain as you move throughout different areas, or alternatively, like from along a gradient or something like that. Yeah.
A question. I know the mesoscale, like, there tends to be a lot of canonical neuroplastic processes that are observed at the mesoscale, like Hadrian processing, Belting, P, proteins, and the like. How would you, I mean, is there methods that you're aware of that you're the ones at fMRI that study these kind of mesoscopic processes at this level?
Some of them. I mean, we can look at things that result in changes in activity. That's kind of our area of expertise when it comes to fMRI. But looking at chemical shifts or changes in the amount of different types of chemicals that would be associated with Hebbian processing or something like that, that's outside of our realm currently. But I think that things are changing over time. Like we are gaining new technologies all the time, etc. But I would say, at least in my experience, the ways that people tend to look at processes that are just a little bit outside of our realm, but we can like kind of get glimpses at through activity, is to build sophisticated computational models and then try to tune them so that they match whatever data that we have, and try to make it so that the parameters that are associated with learning or whatever it might be, some kind of plasticity, which parameters are those that made sense with the data that we collected. So I would say that it's a little bit of an interchange between the people who are doing experimental data collection and those, as you're aware, who are doing like computational modeling at the connectionist level. Does that make sense?
Big person.
Which is not a perfect answer. We would love to be able to measure everything, obviously, but unfortunately, MRI is really good at certain things and it's not always perfect at other things. So that's one area where we need to keep progressing. Okay. We saw it in the last lecture, but I'm just going to repeat it, that one example of cortical columns that people have measured is to look at orientation and ocular dominance columns in visual cortex. Here we can see on the right-hand side these little patches of different colors. Those little patches of different colors are areas that care about different orientations. And then on the left-hand side there, you can see it caring about the right eye or the left eye individually, or preferentially, rather. And this is really interesting because we actually already knew this information. We knew it from monkey studies for quite a long time. So being able to actually collect data in humans that matches that was a huge step forward. I think this has become more advanced over time. Here's an example of being able to look at connectivity between different ocular dominance columns. This is work by somebody named Mariana Schmidt. And what they're actually doing is they're combining both the analyses that are associated with getting individual columns on this cortical surface, and that also the voxels in different ways. So basically what they're doing is they did a mapping experiment where they tried to understand how different portions of V1 respond either to one eye or the other eye individually. And then they tried to understand, okay, if we look at connectivity between these, is the connectivity lower when you're going to a column that is from the other eye? Are you more connected to the ones that are all of the same eye, et cetera? And how does this change through the different cortical layers? And I think that the end goal of work like this, or at least one of the stages, is that this is really important information in the study of amblyopia. And so there's a whole group of researchers that are really, really interested in understanding the responses in different cortical layers and different cortical columns in order to gain more information on how to actually treat amblyopia in the future. We also saw this last week, but I'll just mention that we can, because of the power of ultra high field, measure the responses to individual fingers being tapped in the scanner, and then we can come up with maps like on the bottom right hand side here, where you can actually see all of the different fingers lined up and how they form a gradient. What I think is also really interesting is that sometimes just doing these types of mapping experiments and adding small amounts to the experimental designs can actually give you new information that we didn't know from either from animal studies or from traditional fMRI study. So this is an example from Renzo Huber a few years ago, and essentially they did a very similar mapping structure. Here, they're actually looking at the columns as you move down somatosensory and motor cortices, and looking at which fingers are represented as you move down it linearly. And what they find is in the somatosensory cortex, we saw what we just saw in the other maps, which is that they're lined up in the order on the hand. What we didn't know was that in motor cortex, if you do different types of actions, those are not necessarily the same maps. And so what they found was that if people are squeezing a ball or alternatively retracting their hand against a rubber band, so in other words, abducting or abducting, they actually are different maps along the cortical surface. And what they saw here is that it's actually the hand just in reverse. So you're looking here at one of those representations, and then it's a mirror copy of those fingers as you move back out. But then it's actually important that if you look at which action do they care about, that one of those representations is the squeezing, one of them is the pulling. And so that's new information that we didn't know about how stuff is represented in the brain. Okay, I'm going to shift a little bit from just talking about what people have done to what might you want to do if you wanted to do an fMRI study. So the first thing, and I think it's not necessarily overlooked in people who are doing fMRI, but keep it in mind if you're designing an fMRI study for looking at high resolution, is you need to be really selective with your participant recruitment. As you move more into clinical groups, this becomes less possible because you want to actually study people who have particular conditions, and they often come with different types of behavior. But oftentimes when we start out, we really just want as clean of data as possible to test our experiments. So find your favorite participant. And what we want to also do is try to keep them as still as possible. So in this particular case, you can see that I am taped to the scanner so that I cannot move. And you want to do this in order to reduce motion as much as possible. I don't think it's surprising to anyone that if you get a higher resolution camera, you don't want people to move because your pictures will be blurry. So the same for MRI. The higher resolution you go, the longer things take, etc. So you don't want the pictures to be blurry. One of the best ways that we've found in our center is to use what are called inflatable head pads. These are in both of our 70s. I believe they're also in 3TD. If you ever want to use them, just let us know and we can show you where they are. But essentially what these do is they hold the person in place, but the person has control over how tight they are. When I'm in the scanner, I usually make them extremely tight because one, I don't like to think about not moving, but two, it blocks out a lot more sound than you would have otherwise. There used to be a company that made head cases, and that is maybe even one of the most ideal circumstances because it's like specifically shaped to the person's skull or their hair, etc. And it holds them exactly in place. That company doesn't exist anymore, so people are still trying to figure out how to do this best. But that would be one option if you're really into 3D modeling. And the third thing to keep in mind is talking to your participants and training them to make sure that they know how important it is not to move is an overlooked but really important factor in doing these types of studies. So emphasize to your subjects that when they move, we're losing data, etc. I mean, be polite about it, obviously. We also saw this last week, but I'll just point it out again, that when we shift in our domains to really high resolution work, which is required in order to do layer and columnar fMRI, we shift in the types of sequences that are best for doing that. So in our case, when we're sub-millimeter, we really most of the time want to think about a 3D EPI sequence, unless there are special circumstances that would make it so that 2D actually works a little bit better. And people are welcome to ask questions on when that might be. Also, because when you go to really high resolutions and these images take a little bit longer to form, we're also kind of at the mercy of distortions. And distortions happen because not everywhere in the MRI field of view is actually at the exact same magnetic field. So we talked about it again a little bit last week, but I'll re-emphasize it. When you have a magnet and it is of a particular field and you put an object inside it, that object disturbs that magnetic field in some way. Humans are really, really good at this. And we have a whole bunch of different tissues in our head, so this actually becomes a little bit problematic. If anybody's ever done studies on things like frontal cortex or DLPFC, et cetera, you'll be aware that being really close to the sinuses is really, really, really hard to study. And it's because your field is so disturbed here. So you can see that that's like 60 hertz off of the frequency that you would like to be measuring at. There are a lot of different methods to really try to figure out how to best process this. But I would say the most common one is to use basically running stuff with the phase encoding in one direction and then run the exact same thing with the phase encoding going in the different direction. One of the maybe convenient things about distortions and the fact that they're caused by field disturbances is that if you just flip the direction that you're actually collecting your scans, all your distortions will be, they'll still exist, but they'll be exactly opposite. And so rather than the brain getting kind of splayed out from the front, it might get sucked in. And so then you can use algorithms that basically try to understand, okay, if the brain is here and it's here, well, the real position must have been exactly in the middle, etc. And I'll, again, I've talked about this before, but like, I'll just re-emphasize that when we're looking at cortical layers, which are pretty small little strips of cortex, it's very important that we do these kind of processing streams right, because otherwise you'll be off in where you think layer 1 or layer 4 are. I'll also just point out, this is a method that came out relatively recently from NIH by Kenny. If anybody scans downstairs, you'll know who he is. But Kenny and Renzo worked on a method where they basically took really high resolution EPIs and they ran the phase encoding in every direction possible, so that they could then create an anatomical that could always match your functional data, because you would know what the model of how the distortions were happening in the anatomical. If anybody's interested in using this, it's called T1234, which I always get wrong. It's a T1 scan. It has 2 inversion times. It has 3 different, oh, I wrote it down here. It's 3D, and it's also 4 different combinations of read and phase inputting. So that's where the T1234 comes from. And if you're interested in that method, I can help you get that set up. Okay, so now it's time to layer. If we want to create cortical layers, basically what we have a couple of different options, but I'm going to show 2 of the most common methods, I would say. The first is to use something like FreeSurfer. Those who have done fMRI analysis will be acquainted with FreeSurfer. And basically what this is doing is it's taking an anatomical and it's defining what the surface of that anatomical looks like based on that, T1, sometimes T2. But in the case of cortical layers, you can add on an extra step, which is that it's basically going to find not just one surface, but it's going to create a whole bunch of surfaces in between the surface of the gray matter and the gray matter white matter surface. And the nice thing about this is because it's based on meshes, basically what they're doing is they're taking the exact same vertices and they're just moving them in a straight line that is perpendicular from the surface itself. This means that you have an already built-in version of cortical columns, because basically you just have all these lines that are going directly into the cortex. And I'll also point out the work that I showed where they were looking at connectivity between different columns on between different eyes in V1, that was done using a method like this. Alternatively, we can not think about cortical layers as surfaces, but we can actually just look at the voxels themselves. So this is not looking at the surfaces, but thinking about it strictly in the volumetric sense, which is how we record our data. Yeah. Actually, okay, so I went a little bit out of order here, but basically one of the really nice things in comparison to the volumetric approach of smoothing is that if you have meshes, you can actually define kernels that are based on those meshes. So in other words, you can smooth along the cortical column or along certain layers, but like only take into your local neighborhood, etc. But you can create all kinds of clever ways of processing these data. Okay, what I was saying before. You can also think of your data as like purely volumetric. And so you can just look at the individual voxels and analyze those, but then assign them to particular layers in kind of your showing of results, etc. And this is the approach that is taken by a program called Weenie. This was largely written by Renzo Huber, but also a lot of work done by Bert Vauban in Maastricht. And basically what they do here is, like I said, they take each individual voxel itself, but then they actually are just assigning it to a different number of layers, which they have projected onto the volume. And so you can see they can get layer profiles at different resolutions because they're basically just creating more or fewer numbers of layers in the volume. The other really nice thing about working in the volume space is that you don't have to look at the whole brain, which is not really something that FreeSurfer is capable of. FreeSurfer's algorithms are largely based on the fact that most brains look very similar to one another. And so we can process the whole thing kind of as just one algorithm. But in the volume, you can basically just take different areas. And as long as you yourself can label them as different types of tissues, you can then use their algorithms to turn the gray matter in this particular case into different cortical layers. And you can even create like 3D models of the gray matter in each of your areas in order to better understand their relationship to veins or anything like that. And all of this is kind of a joke, but all of this really comes down to like the really, really important questions, which is if you're reporting your layer results, it's very important to think about which axis are your cortical layers. And there's a huge debate in the field, which is that a lot of people think that the layers should be on the vertical axis, and other people think that the layers should be on the horizontal axis, and then we should show whatever data you have on the horizontal axis. So I'll let you make up your own mind on which is right. I know which is right, but that can be one of our questions. Sorry,
The axis is always right.
sorry,
The Z axis is always correct.
correct for layers?
Yeah.
Okay,
Yeah.
okay. Interesting.
Okay.
Okay, and similar to last time, I'll just point out that there are a lot of resources that you can look for more information and what's like the most cutting edge in these fields. I've gone through a lot in this lecture, and it's a lot of information, I would say. And there's a lot more that you need probably in order to just like hop in and actually do a study like this. So ISMRM is a great place for learning about brand new methods in the layer fMRI and column fMRI space. But there are also two that I'll point out. There's a journal club that meets and basically has people that show their current work every week. It's online. And then there's also layer dinners. Just kind of maybe a personal plug because I often post it. So come see me talk. Anyway. Do you have any questions or anything? No? Okay. Anybody online? Awesome. Well, we finished on time again, so this is perfect. There's only one more lecture in the series, so make sure that you come next week. Peter is going to be talking about some of the biggest challenges and controversies that exist in fMRI and what we can do to try to get at them. Thank you very much.
This, your face, now this.